population ecology session. Bear in mind that people probably will run in from coffee late, so make sure your legs are plastic enough to let them in or move into the middle, so there's space for people to sit down. Check your mobile phones and turn them off. We have one of the speakers that I know of that will either have a no tweet sign, the first speaker, Christopher, on his talk, or please be warned, I've just told you, no tweeting of Christopher's talk. The others say tweet away. So if you like tweeting, wait until after Christopher's talk and go for it. Christopher, do you want to get
I'll probably have to figure that out, I think. Great. So thanks everyone for turning up. First talk of the session. I'm going to chat a bit today about some work we've been doing looking at detecting early warning signals in wild populations. So I'm not going to labour this point too much. You all know the story. We know that biodiversity is being lost faster than at any point in the last 65 million years. I'm going to pick out two graphics just to kind of show this very briefly. The first one is from the Living Planet Index. This was published 2 years ago. And they said that on average, vertebrate species populations are about half the size they were 40 years ago. The second is from a paper that came out last year, who said that the number of species that have gone extinct in the last century would have taken between 800 and 10,000 years to go extinct at background historic rates. So if we all know that there's a kind of a limited amount of conservation funding and effort that can be put towards saving species, and consequently predicting what species are at risk of collapse or extinction would allow us to prioritise conservation effort. And so you kind of really have these two core questions of conservation biology. Can we predict what species are at risk? And thus, are those collapses and extinctions avoidable in the future? And one of the methods that have been proposed to do this are early warning signals. And there is, quite frankly, a bewildering amount of literature on this now. And I'm going to kind of condense this down into 2 lines, which I don't know, some people in here might not necessarily totally agree with. But from my point of view, abundance-based early warning signals have been shown in models and model systems, and in those, they've been shown to do quite well. But when we take that to the real world, I would say they've been shown to be very susceptible to poor quality data, and there's a paper that came out earlier this year who said that they do not reliably predict nonlinear dynamics in wild populations. So we started thinking, well, what are some other measures of stability that we could use to try and bolster these abundance-based early warning signals? And there's quite a lot of work now that suggests that trait shifts, so particularly fitness-related phenotypic traits, can have direct consequences to demography. So shifts in traits such as body size can affect reproductive rates, can affect survival, and those can lead to changes to the demography of a population, and therefore that population's persistence through time. And you might expect if you have increasing pressure on a system, that might lead to trait shifts, which will then concurrently lead to demographic shifts. So actually, tracking fitness-related phenotypic traits might be a secondary way to measure the stability of a population or ecosystem. So we published a paper on this earlier this year, showing that actually if you do this, you can better predict population collapse, and you can actually predict population collapse earlier. But so far, we've only shown this to be the case in small-scale experimental systems. So what do we need to do to take this to the real world? Well, we need to have data on a species where we have body size data of individuals, we have abundance data, and critically data that precedes known population crashes. So the early warning signals we get, we know are true positives. So during a recent visit to IMAS, the Institute for Marine and Antarctic Studies in Tasmania, we came up with the idea of using 20th century whaling data to try and look for these early warning signals of collapse. So the IWC, the International Whaling Commission, have collected an amazing data set. It really is, it's staggering actually. They think they've got every known legally caught whale from 1900 to 1985. So we've taken a subset of that, and that's 4 species, over 2 million individual whales. So these are each individual records of whales caught. For most of these, we have body size data. And so we've taken this, and of course we know that there are large population collapses caused by overexploitation. So we've taken these and we've applied these trait-inclusive methods to these data. So if we have a closer look at the IWC data set, there are 4 species we've picked out. So this is the blue, fin, sai, and sperm whales. And we fitted GAMs to this data. So along the x-axis is year, along the y-axis is the number of whales caught in each year. We fitted GAMs to look at when the pressures on this system are released. So So you can see the black lines here. This is an increasing fishing pressure, and we identified this point as where the fishing pressure starts to subside. So that's either because the populations have collapsed or because they're no longer commercially viable. So for the eagle-eyed amongst you, you will see there's a dip between 1940 and 1945. That's caused by World War II. So whaling didn't actually cease totally, but it reduced quite dramatically. And I just kind of want to take a minute to show you this x-axis and the number of whales that are being harvested each year. These are kind of staggering. So all of these species peak at about 30,000 individuals being removed per year. And for fin whales, you're looking at not far shy of 900,000 whales caught over an 85-year period. And of course, this has some quite dramatic effects on the body size of these whales. So for the blue fin and sei whales, you see this characteristic increase in the size of individuals caught between 1900 and 1930. And we can actually kind of attribute that to a quite cool secondary kind of event, which is between 1900 and 1930, there was a huge increase in the number of patents registered for whaling. So in 1900, whaling was still relatively primitive. But by 1930, you had things like exploding harpoons, fast outrigger ships that were actually able to chase larger, faster-moving whales. And so we see this increase, which we think is an artifact of technological advancement for these first three whales. We don't see it in the sperm whales, and we think that's because they swim about 10 kilometers slower on average than the other three species, so you don't actually get this same artifact. And if we look at the standard deviation of body size of these species, you see this, I think this backs up our kind of point about the technological artifact to some extent. You see again this increase in the standard deviation followed by a decrease. So we believe this is obviously an increase where you're catching larger whales now in 1930, and just the size distribution of the whales you caught are much larger. And then this decrease subsequently is actually, we think, a decrease in the actual size distribution of those populations that you lose the largest individuals. Okay, so I'm going to present three different analyses on this data. We're taking the best metric we identified from our previous paper in 2016 and its component parts. And that best metric was a composite early warning signal based on the coefficient of variation of the abundance data, the mean body size of individuals in the population, and the standard deviation of body size of those individuals. I'm not going to go into detail about how the early warning signals are calculated because I don't actually have time to go over it here. If you want to, I'll direct you to our paper and you'll feel free to read that. So the first analysis I'm going to present is just using all the data prior to the collapse in the number of landings. And that's simply just to see whether there's an early warning signal present or absent, and just an indication whether these systems are becoming destabilized by these pressures. So if I just give you a quick cartoon example of this, here we have the sperm whale data. So we're basically using all the data from 1900 until the year before the collapse, where this blue line is. Okay, so here's the graph. So an early warning signal is present where there's a dot, and obviously an early warning signal is absent where there is no dot. Now, the x-axis are the different species, the y-axis are the different component parts of the early warning signals. Probably not worth reading these. I'm just going to break these down to the three different colors. So early warning signals with an orange dot are based on body size shifts only. The green dots are abundance-based early warning signals only. And the blue dots are abundance-based and body size signals. So if we look at this, you can see that there are body size signals only in two of the species. The abundance-based early warning signals seem to do particularly well across all four species, and the three of the four species produce early warning signals with a mixed abundance and trait early warning signals. But we know that early warning signals are very sensitive to the quality of the data provided. So one kind of important question when trying to apply these to real-world systems is what is the minimum amount of data required to predict a collapse? So we expect the signals to be strongest closest to the time the collapse in the number of landings, because that's when the pressures on the system are the highest. So what we've done is taken, here's our data again, we've taken the 10 years directly preceding this collapse, so we expect the pressure on the system to be highest, the instability in the system to be greatest, and so you'd expect early warning signals to be present here. And we've analysed this for an early warning signal, and then we've iteratively added in additional years and reanalyzed it, and looked to see then what the minimum amount of data required is to produce a true positive signal. We know it's true positive because the collapse happens directly after it. So here is our 10-year initial window, the species down this side, and again, just look at the colours. I'm sorry, the dots are a little bit small, I expect them to be larger than that. So you can see that for some of the species, for the sei and the blue whale, you actually need 10 years of data to get a positive early warning signal here. For other species, such as the fin whale, you need about 20 years worth of data. just the take-home messages from that second one is you can get data, early warning signals with as little as 10 years data, and those signals are present in the size only, count only, and size count metrics. And the previous plot showed that if you include size, you can reduce the amount of data required, certainly in the blue and the psi whale, and critically, I think it doesn't increase the amount of data you need, or the length of data you need in the other species. So the third analysis, I think, is probably the most important of the ones I'm presenting, and that is how reliable are the signals that we generate. And this is kind of essential if you want to put these, apply these methods to real-world collapses. So we know that early warning signals are present in our data, but we also know that all of the whale populations collapsed. So how do we account for false positive versus true positive early warning signals? So the ideal situation would be to have some control populations that didn't collapse that we have data on, but the other whale species recorded by the IWC, the data is really, really patchy. These are the kind of the 4 best species that we have, and so we don't really have a control whale system. So working on the premise that early warning signals should be more frequent closer to the time of collapse, what we do is we take a 20-year rolling window, so that's the maximum amount of time, maximum length of a time series required to generate an early warning signal, and then we roll it through the data. So we take a 20-year window here, we analyze that for an early warning signal, and then we roll this 20-year window towards the time of collapse. And then we say that basically, if you're getting isolated signals, so these ones out here, these are probably more likely to be just vagaries in the data causing early warning signal. But if you get consistent concurrent early warning signals as you kind of move towards this time of collapse, you'd expect that these might be more likely to be true positives because they're reflecting more and more instability within that system. So just to kind of show you the graph of that, so here is the end of the data. So the black vertical lines here are the collapse of each of the species. The blue is our 20-year rolling window. And we can see that you get these consistent concurrent warning signals before the collapse in the blue, in the fin, and in the psi whales, and to some extent in the sperm whales, so that's a little bit earlier on. So this is what we would consider a true positive and reliable signal. Signals such as these are more likely to be false positives, so they're just more likely to be based on vagaries in the data. So conclusions from this last analysis, I would say that reliable signals are produced up to about 40 years before collapse, and that's around 4 generations in sperm whales, although you could probably argue that a couple of different ways. And size count metrics and count metrics perform approximately as well as one another. So the conclusions from the talk in general. I believe we're the first to show, my things have been moved around a little bit, that isn't meant to cover half of the text. I believe we're the first to show that body size shifts occurred in whales during overexploitation, and the sperm whales caught in 1980 were on average about 4 meters shorter than those caught in 1900. And some of those body size shifts occurred before population collapses, particularly the shifts in the standard deviation of body size. Early warning signals predict the collapse in the landings in all 4 of these species, and including trait dynamics can reduce the length of the time series needed to predict these collapses. Again, consistent signals can be picked up around 40 years before collapse. I think given the known reliability issues with count-based early warning signals, traits should improve the reliability of these methods. And just a kind of a quick thought about the future. So there's this paper that was published earlier this year saying that early warning signals didn't predict nonlinear change in wild populations. I think the jury is still out on abundance-based indicators. Some of them have shown to be very positive, some much less positive signals. I think that including traits really could be useful if the data is there, and we've shown that in body size. But you could think about other things like age-sex ratio of a population, the age structure of a population, and these are kind of some of the questions we're starting to think about. But you could also do this for multiple species, right? Data from multiple species and multiple traits from multiple species. So with that, I'd like to acknowledge my co-authors, at least one of whom is in the room. The SNF for funding my trip, IMAS for hosting me during this work, and ERC in my group, and I'll take any questions. Thank you. So I think if you take out mature individuals, you're more likely to dent the population. I don't know that much about whale biology, I would say. Yeah. Yeah, it could be both, I guess, right? Taking large individuals. It's probably taking larger given the technological advancement, I would think, in that you're just able to catch larger whales. They move faster. I think it's probably shifting, yeah.
But in any case, my question is like, so for whales, the issue is overharvesting. How applicable do you think this approach would be to non-harvested species?
Right, so the data we based our original paper on is from a non-harvested species. So it's an experimental population, but we did a bottom-up forcing. So we took away food from the population to force them to collapse. So I think the major difference is that you're likely to get different changes in the standard deviation of body size signals. So if you think about a population that's competing for a food resource, for instance, you're likely to get size-dependent competition, right, for food. So you would expect there to be an increase in the body size, in the standard deviation of body size, which precedes a collapse, which is what we saw in the experimental system. Here, I believe that the decrease in the body size is the critical kind of indicator, because then you're losing the size structure of the population. So that increase that we see is just an artifact of technological advancement, and the decrease is the signal that we're looking for. So I think looking at it in two different ways. For harvested systems, you would change the signal you're looking for. So we looked for a decrease in the standard deviation here, because that's what the fisheries literature would expect you to see. And in maybe other systems, bottom-up forcing, you would expect there to be an increase in standard deviation directly before the crash. Catch me later if you want. Yeah. Cheers.
Thanks, Christopher. Lots of talk for that. Thank you very much. Okay, so next we have Emil Van Loon speaking about forecasts by an operational population model. Thank you.
Thank you very much. Before I'm going to explain why I'm so interested in analyzing this model, some background. The Netherlands is a wet place, and that's why this animal likes it as a habitat as well. It can lead to accidents. This is a recent dike breach in Italy with consequential flooding due to this kind of digging. So this muskrat is a semi-aquatic rodent native to United States and invasive in Europe. And these are some of its ecological properties or traits, if you like. And it has been invading the Netherlands since the 1940s, and since then the catch has been well recorded. And here you see the development of the catch over time. And after initial invasion in the south and increment towards almost the entire country, it started to decline later on. And apart from the catch, also the effort has been recorded quite systematically, except for some, as you see, some lacking periods. And it is on the basis of this data that I was involved in the construction of a spatially distributed model, which predicts like not the catch itself, but the population density or the population numbers in the country. So this leads to predictions like these, maps basically. And the interesting part of it is it doesn't only give maps, but it gives maps with confidence intervals. In this case, I show you the time series of the prediction. Not in the black line you see the catch itself, and in the red line the predicted population size. But also some interesting features like the fraction that's being caught with kind of confidence interval, at least a first order estimate of it. And it's and this is a pseudo, to give an impression of the kind of predictive power. If you translate it into pseudo R squared, you have to think about 0.7 kind of accuracy. So why I'm interested in improving this model, there are two reasons. First of all, it's a fairly general population dynamic model with a spatial component in there. And if anything works on this kind of model, it works on a large family of models. And secondly, this particular model is used operationally. So there's this organization that has to catch these animals is very, very interested in how they can bring down the density with as least effort as possible, and with as least cost as well as possible. So I'll quickly sketch 3 properties of the model so that you can understand the interpretation of it a little bit better. It consists of 2 classes of animals, so to speak. There's adults that can have their death, the D, and can be caught, the C, and remaining survives. And they have offspring. These are youngs. And these juveniles, they are juvenile over a year, and at the end of the year, they enter the cohort of adults. And so there's a birth rate in there, and there's a spatial exchange. So in space, this animal also spreads every time step. And if you put this equation into this model into equations, and you see here the colored parts are most interesting. So in yellow and green, for example, that. And as you see, these coefficients are equations, kind of most of the time nonlinear equations of some environmental covariates that were available for this study. So these aren't static, but they change in space and sometimes also in time. So to summarize, so in its basis, this is a very standard model, but there is some complexity, mainly due to this nonlinear relationship with environmental covariates and due to its size, because we have these 5 by 5 square kilometer blocks, so this makes it a big model with a lot of state variables, if you like. Now something about the estimation method. If you run this model without doing anything apart from initial conditions and then say go, strange things happen. That doesn't work. I call this an open loop simulation. It would be something like the blue line. So in order to have sensible output from something like this, you need a correction, and that's why this is a state correction approach, also called filtering. At every moment that you have an observation, like here we have the blue line predicting catch, and the green line with these strange jumps in there also predicts catch, but then the model is adjusted each time step that you have an observation. So it stays closer to these observations. And these jumps aren't really nice, but you can play a trick on the system by running the filter backwards again, and then it's called a smoother, and you get this, for example, this smooth red line. And this procedure of adjusting the model each time step that you have an observation towards the observations is called state adjustment. And to give you an idea how this would work in this context where you have catch, which is in fact an output of this model as well. I put there a box in this figure to denote this. It's also, the actual catch is also being realized. Here it is. And it is this particular mismatch that is fed back into the model and translates through these other red arrows into the other variables of the model. And then, as a matter of speaking, the residual or mismatch in the catch translates to a mismatch in these state variables in which you are really interested. So it's this propagation of the residual which is the handle that we'll use, or the thing that we need to understand in order to interpret the model output. So now I will simplify things a little bit, starting from this picture that you saw before. Let's leave out a few things, and now I'll put back the influential variables at the right, and the state variables that we're interested in, the number of adults and the number of juveniles at the left, and the crucial functions, functional responses, if you would like, given in the middle. And these arrows give more or less the influences that were put in the model in the first place. And so one thing you can do is to select one particular individual relationship in here, like the one that I'm going to show now, from habitat towards the number of adults and juveniles. And so the residual of this model, the degree at which it was corrected, is put on the y-axis. And the effective variable that was used, a very simple variable, soil type is the most effective one for this particular animal, put on the x-axis. And here you see that, strangely enough, there is indeed this is an overestimation of the number of individuals by which this model has been corrected, has to be corrected each time, and there's a structural under-representation here. And there's a small difference in between the blue, which are the young, and the red ones, which is adult, which is systematic as well. So, this can readily be interpreted as that there need some changes need to be made in these response functions. Similar to this one is, for example, if you put the difference in population size between the focal spatial unit and its surroundings, that should drive the diffusion of this animal into the landscape to surrounding cells. And if this difference between a cell and its surroundings is negative, then you get different errors from when it's positive. It's quite asymmetric, something you wouldn't expect. So there's something definitely wrong with this kind of diffusion parameter. You would like to in fact, see these lines along the zero axis, the zero value here. So next idea is to put these variables in a model in combination. And the obvious choice then is multiple regression, which I did, by which not only these variables that I did show in the previous pictures, but also a few others, like density itself, effort, are all related to the residual. So this model, a linear model, is not made to analyze real data against environmental data, but to analyze model errors against the predictors that we have at our availability. It gives us some leads, but I think rather than to try and interpret these things, it is the main conclusion for me is that it's rather difficult to translate these things directly, because there are still these intermediate variables which are in fact doing the job, and there's no way by which we can estimate these latent variables with multiple regression. So my next step here was to try and formulate structural equation models, and these intermediate variables, which are also called latent variables, which we cannot observe directly, but we can observe these, and we can observe, at least from the model output, these can be analyzed with these tools. And what these tools show, I did only show the significant correlations in this graph, show which of those predictors or environmental covariates relate to which intermediate variables and how these translate into the residuals. So there's one here for adults, there's another one for juveniles. I think the actual values are perhaps not the most interesting one. Here's an explanation of what let me point you at this thing, how I was pleasantly surprised to see, I have to go to this one, to see the effective habitat there being stated as that something had to go down there, at least if the habitat was peat soil. This structural equation model identified that this bird was too high and had to go down. And that was exactly what, when digging back into my original functions that were formulated in the model, what seemed to make sense, since peat was indeed, on the basis of earlier literature and field experiments, set as a higher productive area than the other soil types, and by lowering it by 0.15, it gets almost as high as the other soil types in there. So along this way, there are other response functions that are really difficult. This is like piecewise linear relationships that I don't know yet how to change, in fact, on the basis of these structural equation models in a systematic way. But so far, these are my conclusions. I've attempted to dig deeper into the forecast of the population model. Some things I didn't show you, like mapping errors in geographical and environmental space, which is insightful, but doesn't lead to new proposals for new models. Multiple regression also, not really. Structural equation model seems the most helpful until now. And the preliminary results, they point at a lack of density dependence in the model as it is now, which information we couldn't get from the literature, and information that many people said they should be in there, but we couldn't get any handles to formulate them. And the variation in catch efficiency, for which we don't have a good predictor variable, but the errors remain high on that one. So thank you very much for your attention. And I'd like to finalize by thanking my collaborators and then take any questions from you. Thank you very much. Yes, every 5 square kilometer. That's why it's, otherwise it wouldn't be possible to fit such a thing, yeah. Go ahead. The catch data, can you repeat? The catch data are the results? Yeah? Yeah, so you can enter in. I try to simplify it as much as possible to get through it in the time window. So there's like an intermediate step. Yes, there is effort number of hours that are being spent, and the population size of the previous time step. So every time slice is isolated and that's kind of a boundary condition. So this catch is in fact already pre-calculated from a basic catch per effort parameter times the effort times the number of individuals. Yeah, you're right. So by picking that up.
So you showed that this is a problem, and you said that there was a certain catch. But the catch of the population in terms of tracking relative to the total population size is tiny. I mean, in terms of if you were able to find a variable that's able to be controlled to assist in the control of the muskrats or the management of the muskrats, is there one variable that you've identified that actually helps you more than others in the management?
One variable that I can observe, you mean? Well, no, but I think that not yet. I think that knowing a lot more about the dispersal of this animal in its migration or diffusion period would be very, very helpful, and for which there's small experiments ongoing to measure that better. And for the rest, no, I don't think we know that already. Thank you very much.
Thank you very much. Thank you. It's not you. No. You have that on the list as next.
This is what happens when you're nervous about. No, what, me?
Yeah, you're
Yeah.
next. You're next after this talk.
Yeah, this is what happens when you're nervous.
It's up to you.
I can give your presentation if you want.
Okay.
So thank you. I'm Marlène Gazon, and I will present to you a work entitled The Predictive Framework of Climate Effects on Population Dynamics, going beyond population size. So predicting climate effects on population dynamics has become urgently required of ecologists, and is also crucial for producing appropriate management and conservation strategies. However, this task has remained a challenge due to its scope, because climate effects can differently influence the vital rates, the survival rates, the recruitment rates, and the immigration rates. Climate effects can differently impact the stages or the ages of the life cycle. For example, in a social population, it has been shown that young survival was mainly influenced by North Atlantic oscillation, whereas the survival of older individuals was mainly affected by rainfall at the end of the winter. And finally, climate may interact with density-dependent factors to affect the population dynamics. Again, so for example, when weather conditions influence some limiting resources, we should take into account both climate and density. So again, in this social population, it has been shown that bad weather at low density has no effect on overwinter mortality, whereas the same bad weather at high density may induce important crashes in the population. So considering these interactions between climate and density is really crucial for producing appropriate management scenarios. As a consequence, reliable models of population dynamics in a changing environment must include such interactions between density and climate on all vital rates, survival, recruitment, and immigration rates, and also at all ages or stages of the life cycle. This bird, the dicker, is a small passerine bird, widely distributed at northern latitudes. This bird depends on open water for foraging and running water for nestling. So clearly, during the winter, the amount of ice will influence the availability of feeding and breeding habitats. Previous works, based on a population located in Norway, have highlighted that the growth rate of that population depends on climate conditions, more specifically on winter conditions, and also on density-dependent factors, meaning that the larger the population size, the lower the population growth rate. These studies have concluded that under global warming, we should expect an increase in mean population size for that population. But none of these studies have explored yet responses to changes in climate. So here we develop an integrated framework of climate's effects on population dynamics, including interaction between density and climate on all viterates and for all ages. And based on this integrated framework, we will project the future persistence of the population using local climate scenarios. So as I said, the population is located in Norway. Breeding sites are concentrated on the banks of the river. Between 1978 and 2013, occupied nests are recorded, fledglings are ringed, and ring mothers are identified. So basically, we have access to three different kinds of data: capture, recapture data, population counts, and reproductive success data. Unringed mothers are given a ring and are assumed to have immigrated into the population the current year. In total, 992 breeding females of non-age, local plus immigrants have been monitored. So in this study, we consider four different age classes. So briefly, the first age class here brings together females locally recruited and immigrants, because most of the immigrants are females of age 1. These individuals may survive and enter into the age class 2 the next year. Females of age class 2 may survive and enter into the age class 3, and so on. And individuals from age 4 may remain in the same age class if they survive. And all these females are able to reproduce. So we estimated survival rates for each age class, recruitment rates, and the number of females in each age class each year using a Bayesian integrated population model, analyzing reproductive success data, population counts, and capture-recapture data at the same time. And then we explored whether survival rates may be defined as a function of density and climate. And for climate, we consider winter temperature. So you can see here the survival rate for age 1, for example, and here the density, so the number of females in the population. Each line corresponds to climate conditions during the winter. So basically, the black line here corresponds to cold winters, and the blue line to mild winters. And you can see that warm winters increase survival rates, so the survival rate is higher at warm winters than during cold winters. And you can also see that the responses to density dependence depend on climate conditions. So there is a strong density dependent response during cold winters, and there is a weak density dependent response during warm winters. So basically, we get exactly the same pattern for all ages. So you have here survival of age 2, 3, and 4. And you can see here, so again, the cold winters and the blue line mild winters. And you can see again that warm winters increase the survival rates. And you can see that the responses to density dependence depend on climate conditions and are really strong during cold winters and very weak during warm winters. So then we explored whether the recruitment rates could be defined as a function of density and climate. So again, here you have the recruitment for age 1, and here the total number of females in the population. Again, the black line corresponds to cold winters, and the blue line to mild winters. And you can see that warm winters increase recruitment rates, and that the responses to density dependence depend again on climate conditions. So there is a strong density dependent response during cold winters, and very weak density dependent response during warm winters. So we got again exactly the same pattern for all ages, recruitment of age 2, of age 3, and of age 4. And finally, we explored whether the number of immigrants joining the local population could be defined as a function of density and climate. You can see here the number of immigrants, and here the density, the total number of local females in the population. Black line corresponds to cold winters, and the blue line to mild winters. And you can see that warm winters increase the number of immigrants, but this is the case only when the density local birth is low. When the density local birth is high, you can see that whatever the winter conditions, you have the same number of immigrants joining the local population. population as a function of density and climate. We have defined the recruitment rates and also the survival rates as a function of density and climate. So basically, we have a population model now that depends on climate and density. So that means that for given conditions of density and climate a given year at time t, we should be in theory able to predict the number of females in each age class the next year. So if it's true, that means that we should be able to reconstruct past population size based on past observed climate conditions. So let's see. If we consider the number of females in each age class in 1979 and the winter temperature in the past between 1980 and 2013, we are in fact able to reconstruct past population size quite well. You can see the thin line here corresponds to observed population size and the thick line to predicted population size. And you can see that there is a quite good match between predicted and observed population size in the past. If we look further into this, you can see that we are able to reconstruct the number of immigrants quite well. And if we look at the number of females in each age class 1, 2, 3, and 4, you can see here the observed values and here the predicted values. And you can see that, again, there is a quite good match between observed and predicted population size in each age class. So that means that if we consider here climate scenarios for the future, we should be able to project the persistence of the population. So let's see, if we consider the number of females in each age class in 2013 and 40 different local climate scenarios for the future, you can see that we predict an increase in mean population size. If we look further into this, you can see that the number of local birds will increase, should increase in the future for all ages, meaning that the recruitment rates and the survival rates would be improved in the future. And you can see that for the number of immigrants, we will reach a kind of plateau. So that means that warmer winters increase survival and recruitment for local birds and reduce the strength of density dependence. So that means that during cold winters, the water is frozen, so the resources are really limited for the birds, and there is a very strong competition among individuals to get access to the food resources. But when the winters become milder, the resources become available, and the competition among birds is weak. So you have seen that there will be an increase in the number of local birds, but in turn, that will reduce the proportion of immigrants joining the local population due to density-dependent feedback. So we should expect an increase in mean population size. If we come back to this figure, which is basically the same for all ages and for all rights rates, that means that we will move from year to year. But you can see that at low density, the population is kind of buffered against cold winter, meaning that a cold winter will have very little effect on survival rates. But at high density, you can see that the population is less buffered against poor environmental conditions, and that a cold winter will have a dramatic effect on survival and recruitment for all ages. So basically, warmer winters should make the local population likely less buffered against occasional cold winters. So I would like to thank all my co-authors mainly from Norway, and I would like to thank you for your attention.
Thank you very much. And any questions for So the question was whether the model had considered the carrying
Yeah, so today I want to talk about how we design monitoring programs and asking this question, how much effort is actually enough to infer that a species is not present at the site? My motivation for this talk came from a news story a couple of months back whereby a property developer was arguing that people were planting newts at sites to halt development. His evidence for this was saying that surveyors went to the sites and there are no newts, and then they look again and there they are. Surprise, surprise. I doubt this is a surprise to anyone in this room. However, this got picked up by lots of news outlets, and I think it highlights two quite important points. The first is that we're often not very good at communicating the limitations of the monitoring that we do, particularly to people that end up actually using the results of that monitoring, so people like land managers and property developers. And secondly, that this misunderstanding can actually have some quite serious consequences. So this property developer is actually also a lord in the House of Lords and was using the platform there to actually argue that protection should be lifted from this species. So there are actually some implications of people not understanding this or us failing to communicate the limitations properly. So the situation that we typically have when we want to monitor species is you have a set of sites that you want to go to, and you realize you have to go there multiple times. Say you choose to go there 3 times, and for some sites, you go there, you visit 3 times, and you see your species there. Great, you know it's there. However, for these remaining sites, you want to know whether or not those sites actually are missing the species, or whether you've just failed to detect it there. And say after 3 visits, you decide that's not quite enough to actually, for me to be sure that the species isn't there, you could decide to do some more sites, some more visits, and maybe some of those sites you find previously undetected populations. And in theory, at least, you could carry on indefinitely until you get enough confidence that you've actually detected all the sites that are occupied by your species. However, in practice, of course, there's a trade-off here. There's an opportunity cost. The more visits you carry out at each site, the less sites you can actually survey because budgets are limited. And so there's a need for kind of a framework to help balance this trade-off and try to survey as many sites as possible, but with sufficient confidence that you're not actually missing populations. And such a framework was developed in the early 90s. And basically, this framework requires one piece of information that you use when you're designing your study, and that is detection probability for your species for the survey methodology that you plan to use. That could come from either from pilot studies or some other source. And then you plug that into this equation on the left here, and then you can calculate the probability of getting a sequence of n non-detections at a site, given that the site is occupied. And this gives you this curve here. And you can flip that around and set a probability that you're willing to accept. So that then turns into this alpha 1 here, say 5%. And then you can calculate n star, which is the number of non-detections at a site required for the probability of getting that many non-detections to be so improbable as for you to be confident to say the species isn't there. And then that n star should be the survey effort that you invest at each one of your sites in the survey that you go on to do. This is nice and simple. It requires very little information before you do your monitoring. And as such, it's been used in hundreds of monitoring programs to design their studies, and there are dozens and dozens of papers that use this as the basis of their monitoring design. However, it's a simplification. You see this probability here is conditional on a site being occupied. So basically what this n star is, it assumes that if you don't see a species, it's because it's hiding. And if you see a species, it's because it's there. But obviously that's not the only possibilities here. In fact, you could get lots of non-detections at the site because it's just not there at all. And really, this is what we want to figure out when we're doing species monitoring. So this took quite a long time for people to realize, but a few years ago, a couple of papers came out that adjusted those equations you saw previously and incorporated an estimate of species prevalence before you do your monitoring, and came up with these equations here. So they're a little bit more complex because they use this second piece of information, but they effectively estimate a similar thing, except for here the probability is the probability that a site is occupied given a string of non-detections. So it's no longer conditional on the site being present. And again, the n star I've called it alpha 2 because this probability is slightly different, but it does exactly the same thing. So we've got these two different frameworks that people can follow to design their monitoring study. A nice simple one that requires very little information, and a slightly more complicated one which better represents reality. And I wanted to see whether these these frameworks diverge, or whether they're quite similar in the recommendations, how your study would look if you actually followed one or the other of these frameworks. I wanted to do this with real data, so I got some data from Switzerland. For 12 reptile species, there were repeated surveys done at nearly 300 sites across Switzerland. And from that, I was able to fit occupancy models and get an estimate of species detection rates. So this is the first piece of information required for both of these two different frameworks. And then to get the prevalence estimates for the second framework, I went back to a database that exists in Switzerland that contains all species records for the last 40 years. And for each species, I calculated the proportion of grid squares which represent suitable habitat for that species, which contained at least one record of that species. And I did that at 3 different grid sizes for reasons that I'll come to in a minute. So I've got these two pieces of information, plugged them into those equations I showed you previously, and I'm just going to quickly show you a few representative examples to see how the two frameworks suggest different survey effort requirements at sites. So first, for the species on the left here, the snake species, it's quite a rare species. It's not present in very many grid squares, and it's also really hard to detect. And these curves here are similar to what I showed you on the first slide, and what we want to focus on are the vertical bars here, which are the N stars. And we can see that the one-parameter framework suggests that you need to go to each site 20 times if you want to actually be confident that that site doesn't contain that species. Whereas if you actually account for the fact that this is a really rare species, you only have to go there 4 times, and then if you haven't seen it after 4 visits, it's probably because it's not at that site rather than you failed to detect it. For other species, for example, the slowworm here, it's much more prevalent through Switzerland, and it's got slightly higher detection probabilities. And here you can see that no matter which framework you follow, the recommendations are pretty similar, kind of 8 or 9 surveys at each site, and if you don't see it, then you can give up on that site. It's important to note that this is still probably more surveys at any given site than any monitoring program is realistically going to be able to invest, but nonetheless, these are the recommendations. And then if you go to common species and highly detectable species, such as this green wall lizard, we can see, again, the recommendations for both surveys suggest, both protocols suggest similar things. But interestingly, if you take into account the species prevalence and that this is a very common species, the 2-parameter framework suggests that you should just go visit as many sites as you can, go there once, and if you don't see it after 1 occasion, it's probably because it's not there rather than because you failed to detect it. So we can see that for some species, highly detectable species or common species, these frameworks suggest kind of similar survey effort, so there's not that much in it when you try to choose between the two, but for other species, the differences are quite large. However, I really wanted to delve into this 2-parameter framework because this requires information before you start your monitoring on how common the species is likely to be in your study area, which I think in most cases, you're not going to have that information. That's why you want to monitor the species in the first place. And this, I thought, could be slightly problematic because the information that you might have is probably quite patchy. You might have some idea from previous work, or you might just make an educated guess. Or, like I did in this study, you might have some information that you can use to kind of estimate species prevalence. But it's really difficult to actually assess what scale you should be looking at. There are very few species for which the natural scale at which you can assess prevalence is obvious. Most species live in a big matrix of habitat. And the importance of that is when you're designing your monitoring is, say, this is a study area and these dots are presence records you have. The presence you're going to end up with depends entirely on the grid size that you use. So, for example, using grid squares like this, you could end up with this prevalence. If you use slightly different grid squares, your prevalence is going to shoot up. And again, if you use even larger grid squares, it's going to appear that the species is everywhere. And so if you actually make some inappropriate choices or use data because it's there rather than even if it's not actually appropriate, you can really end up with some quite drastically different recommendations from this two-parameter framework. So we can see for the sand lizard on the left, actually it doesn't make that much difference. This is a common species that's found almost everywhere. But for rarer species, again, for example, this smooth snake, if the recommendations vary from 0 visits per site to nearly 20 visits per site, both of which are unfeasible. But really, this highlights that unless you have accurate prevalence information, this framework might not be the best to follow. And this estimate says you shouldn't visit at all because this species is so rare if you assess it at this small grid size that basically you're never going to be able to put enough effort into any given site to be sure that that species is not there. So you should not bother at all, and you can't monitor that species at all, which is obviously not something that most people wanting to do monitoring are going to want to hear, but it does highlight quite an important point in that for hard to detect species, we're barely ever going to be able to invest enough input into surveying to actually not miss a large proportion of occupied sites. And there are a few things we can do about that. If the species is hard to detect because it's not very common throughout the environment, all you can do really is try and restrict the scope of your monitoring. So basically increase the expected prevalence within your study area. And for hard to detect species, really what you need to do is go down and change the actual methodology that your surveyors on the ground are doing to try and boost detection probability in your individual sites, and therefore carry out fewer, more accurate surveys, which are much less likely to miss populations, leave them undetected, than a large number of visits with low detection probabilities. Again, with prevalence, I think we rarely have useful or reliable information before you start monitoring, so it might not actually be best to follow a framework that uses this information unless you have a reason to believe that it's reliable. And just to bring this back to where I started, again, this issue of communicating the limitations of surveying, I think, again, there's another argument for actually not using this two-parameter framework unless you have good prior information on expected prevalence, because in the one-parameter framework where we started off, this alpha term, which you set as the confidence that you want to have, it actually represents the maximum proportion of populations that you're willing to miss. And this is very easy. It's a number you specify, and it can be plugged into decision-making frameworks for development for whatever, and people are happy understanding that. However, if you use this expected prevalence and you get it wrong, then the equivalent parameter, alpha 2, you could actually end up failing to observe much more than this proportion of the populations, which could be really problematic and really undermine people's faith in monitoring if then later they go and see the species that you said is not there in the site. And that's how you end up with quotes like this. Finally, I want to say that these curves are really useful both to actually see the power of monitoring that we're willing to do, the effort that you can actually carry out at a site, see what proportion of populations you're likely to be missing, and also communicate that to others. And I think that's the only way that we're going to change quotes like this, maybe at some point in the future, to something a bit more like this. And with that, I want to thank my supervisors and people collecting the data, and you for listening.
Thank you very much, Simon. Couldn't be a more important talk at the University of Liverpool. If anyone knows wants to know what I mean, speak to Stuart Plaistow at lunchtime about his love of great crested newts. I'm afraid we've run out of time for questions, so you'll have to catch up with them afterwards. So, the next speaker is... Okay, so Simone is going to speak to us about a focus on genetics project on understanding the structure of natric bats across Northern England.
Hiya, I'd like to start off by thanking my sponsors and my supervisors for all the help they've given me. and to talk about a chapter of my thesis, which is to do with genetic structure of bats. So firstly, why are bats important to us? Well, they represent a large degree of biodiversity. In fact, one in five mammals are bats. They're important pollinators of over 500 different species of plant, including plants that produce that we get tequila and chocolate from. And also another little known fact is that they're really important in the regeneration of fragmented forests. So when larger mammals have moved out of areas, these small mobile mammals can remain and then disperse seeds between the patches, effectively creating habitat corridors. As Chris spoke about earlier, though, we can't just consider biodiversity in terms of how important it is for us. When policy is concerned, we have to think about monetary value of biodiversity. This video here shows a large number of Mexican free-tailed bats emerging from a cave in Texas. And the adult females can eat up to 2/3 of their body weight per evening in pests which are important to the cotton industry. So in the US alone, the value of bats is estimated to be at least $3.7 billion a year. In the UK, all of our bats are insectivores, and although they don't roost in such large numbers, they probably have a massive value to our economy as well. So I'd like to introduce now the concept of One Health, and this is an internationally important movement that aims to bring together vets, doctors, wildlife health practitioners and scientists, because it recognises the fact that bats, animals and their ecosystems are all interconnected. And it aims to tackle problems to do with this, such as infectious diseases. In fact, we've found that about 6 out of 10 of all infectious diseases are transmitted from animals to humans, and this seems to get worse as we further alter the environment. These are a number of bat diseases that have recently made headlines, and all of these are thought to be attributable to habitat alterations. If you consider the case of Ebola in West Africa, the outbreak there is thought to have originated from a bat. And we know that there was large-scale deforestation in the area, which has led to once remote patches of forest now being connected by roads to urban centres. And it's thought that the increased accessibility for people into the forests for hunting, along with the disturbance and displacement of bats, has led to this outbreak. So what about the UK? Well, in the UK, bats are protected under law, transcribed in the EC Habitats Directive. And so it might be more important than ever now we're leaving the EU to ensure that we've got enough knowledge about their behaviour. At present, we don't have basic information about a lot of UK bats' biology, ecology and behaviour. So my work at the moment is kind of strategic research in order to inform those things. There's three areas I'd like to discuss today. Firstly, I'd like to try and describe a natural roosting population of bats. And this will be done in the form of a network diagram. Then I can look at how disease might travel within the population. I'd also like to discuss how the populations of bats in the UK are structured, whether there's local populations or regional ones. And then I'd like to discuss genetic differentiation between sites, which might later be used as a proxy for disease transmission. So this is my study species. It's a Natterer's bat. The UK has quite a widespread population, but we have an internationally important population, and we've got a legal obligation to protect that. And in the EC Habitats Directive requires us to achieve or maintain what is known as favourable conservation status. But to do this, we need the basic understanding of the habitat use and population numbers, which we just don't have right now. Another reason is down to the Bocolo bat lyssavirus, and lyssaviruses cause rabies in humans. And since this discovery of Bocolo in 2010, it's only been found in Natterer's bats, which suggests that they're a reservoir host for this virus. Although Bocolo hasn't been found in the UK yet, other lyssaviruses have, although they're in relatively small numbers at present. So to get a better understanding of bats in the UK, I had to firstly collect some, and I had 5 different study sites. You can see here there's 3 in North Yorkshire, there's 1 up in Northumberland and 1 in Lancashire. The red circles represent known swarming sites. I should mention here that bats return to their summer sites each year to have pups and raise them, but in the autumn they go to big swarming sites where they exchange genes and then later hibernate. So these are the two known swarming sites, although there's likely to be many others. I caught bats in the standard manner using mist nets and harp traps, and I then attached 4 arm rings to enable me to identify them, and radio tracked selected individuals to determine their habitat use and also to find the natural roosts. Out of all of these sites, the only one that was entirely natural roosts is the one in Northumberland, which is up here. Once I found the roosts, when it was possible, I caught from roosts, although as you can see the picture here, in the natural setting it was quite difficult as the trees were largely ancient or very old trees, and the roosts were about 15 metres high, although I did succeed in catching about once per week from roost there. All the bats, naturalist bats that I caught, I took wing biopsies from under licence from the Home Office, which were then stored in ethanol at -20. I used an extraction kit to remove the DNA, and then I used microsatellite analysis and GeneMapper to genotype 152 individuals using 15 different markers. So the radio tracking of the bats at Wallington resulted in, I radio tracked 29 individuals and I found 23 different roosts, which is quite a large number of roosts for a small population. There was a high amount of roost switching, so the bats moved around every 2 to 5 days. And this has consequences for ecological surveys. As Sam was talking about, at the moment when we do planning applications, people will go to a building and they maybe do 2 or 3 surveys. But if bats are having like 20 different roosts that they're using, it may be that you're missing the bats when you do those surveys. The relationship between bats and their roosts, oh, it's not coming up, but there was another picture there, can be described as a roost network, which you can't see here, but it's basically nodes. So the trees would represent nodes, and then the bats would be around those nodes. And looking at central nodes of the roost, you can then predict how disease might spread within the community. And then the central nodes would be highly connected as well, so you'd then be able to look at social hubs and model various different scenarios, such as what might happen if you remove a tree from a network, or how many roosts a community needs to survive. So I used my roost network to come up with a matrix of how often bats shared roosts, and this was represented between 0 and 1. So the value of 0 is when they never share a roost, and 1 is when they're always found together. I compared this to a genetic distance matrix. So if the bats were genetically identical, they would have a value of 0, and if they're more different, that would be higher. I looked at the correlation between these two, and although it was slight, it was significant, and there was a negative correlation, which suggests that bats who roosted together were more genetically similar. In summary, the findings of the high number of roosts and our ability to analyse the network will add to our basic understanding of bat biology and roosting habitats, and we'll be able to further analyse the network to understand better how disease might spread. I then went on to consider how the populations are structured to look at whether they're local, regional or national, and I used a Bayesian cluster analysis program called Structure to do this. If you look at the top diagram here, you can see that the cluster analysis found the likelihood of about 4 clusters. And if you look at the diagram here, you can see the 4 clusters are represented by different colours. So I've loosely grouped them with the white lines. The first cluster is mostly pink, and that's these 2 populations in Harper and Wintringham. So it's likely that they're very similar and perhaps represent a single population of bats. Whereas this one, which I hypothesised earlier would be similar to those 2, is actually quite different and perhaps more similar to the one in Northumberland. So these 2 may share a swarming site. So in summary, I suppose cluster analysis shows us that some sites that are close together are quite similar, although some sites close together are also quite different, and it highlights the importance of the swarming sites. I then went on to look at how much genetic diversity there is at sites, and this can be calculated by heterozygosity. So 0 heterozygosity would represent discrete isolated populations, whereas high values close to 1 would indicate complete mixing between populations. I found a relatively high genetic diversity across all of my sites of about 0.78, and that's similar to the other studies on Myotis bats. It's a good sign in terms of conservation because high genetic diversity enables species to cope with environmental variability and adapt to environmental change. And then I went on finally to look at the differentiation between sites. And this basically looks at how much mixing there is between sites. So higher values of genetic differentiation mean less mixing. I found a mean at our sites of 0.044, which means there's a good degree of mixing. And this is probably likely due to bats exchanging genes at swarming sites. So the figure here illustrates this level of mixing, whereas thicker lines represent more mixing. You can see, again, the Wintergreen and Harpham sites have quite high degrees of mixing, whereas the Harpham and Locaton have quite low degrees of mixing. And perhaps the Locaton bats are going to a swarming site similar to the Y-collar bats. It's very difficult not only just to radio track bats in the first place to find their habitats, but also if you wanted to track them long distance to swarming sites, it's almost impossible without the use of light aircraft because they fly about 5 meters a second. So at the moment, we'd have very little information about the swarming sites of bats, which obviously needs more work if we're going to maintain the high level of mixing. In summary, I'd just like to finish off by saying that the high roost switching of most UK bats needs further attention, particularly in terms of planning proposals if we're going to meet the expectations of favourable conservation status. The 5 different sites in northern England support about 4 different clusters of bats. However, there's a large degree of mixing between these clusters, and there's high levels of genetic diversity, which suggests gene exchange at swarming sites. Again, we need to do some further work to be able to enable us to identify these swarming sites and protect them. And finally, in terms of One Health and disease, populations of natterers are structured at large scales. Therefore, disease might be considered to operate at regional or national scales. Therefore, it's really important that we protect the habitat of natterers, their swarming sites and their roost networks in order to prevent disease spillover from them to humans. So I'd just like to say thank you for your attention. I guess you could, but I suppose those areas would be quite large, and so you'd then have to go and try and figure out exactly where they are. But that would be a good starting point, definitely, yeah. Mm-hmm.
You might be able to detect at sites that you already know because you've got some radio tracking. I don't know how long it lasted, it's more than 4 months, probably not. But what about the GPS technology now? It's getting smaller, we're seeing it on site. So I mean, can we use that to find not all swarming?
I guess with larger bats, such as noctule, you might be able to use really tiny GPS, but with bats such as natterers and Myotis species, it's just not small enough yet to use the GPS technology, unfortunately. We suspect that they do, and there has been a study by other people. Nicola Rivers did a study on recapture at swarming sites, and she did catch a few bats, less than 10, over multiple years at the same swarming site, so it is quite likely. Although there's massive numbers of bats at these swarming sites, so the likelihood of recapturing one is quite low.
So they come to the swarming sites in the spring or summer?
In the autumn, yeah.
In the autumn?
Yeah.
So I was trying to think when people would be most likely to be out, so
Yeah.
trying to keep them out of there.
Yeah, I mean, once they're identified, I don't suppose there'd be a lot of people going into caves in the winter. I think caving is more of a summertime activity, so it may be possible to protect them. Thank you.
Okay, so our next speaker is Sonia Mitchell. She'll be talking about the
All right. So I'm a DTP student from Glasgow University, and before I'd start, I just want to acknowledge my supervisors, Richard Reeve, Louise Matthews, and Christina Cobalt, for their advice and support, as well as everyone else for their contributions to this work. So I'm going to start off by giving a brief introduction to the diversity framework and follow that up with a number of case studies, starting off with a population demographic study of England and Wales, then going on to look at biodiversity in the Barro Colorado Island forest dynamics plot, and then looking at antimicrobial resistance in terms of sequence data, phylogenetic trees, and phenotypic profiles. So our framework of diversity incorporates many well-known measures, and these measures are typically partitioned into alpha, beta, and gamma components. That is, the diversity within a subcommunity, between subcommunities, and across a meta-community as a whole. Our framework is different in that we can calculate alpha, beta, and gamma components at both the meta-community and the subcommunity levels, as well as looking at a range of related measures that allow us to look at subcommunity structure from different perspectives. And we can incorporate measures of similarity that allow us to look at functional diversity, phenotypic diversity, and so on. This framework expands upon a previous measure that was developed by Tom Leinster and Christina Cobalt that calculates the similarity-sensitive diversity of a single population. Further information is available online on the archive paper, and the theoretical underpinnings of this framework will be expanded upon tomorrow by Richard Reeve at 1:30. So to get a better understanding of this framework, we start off with a relatively simple data set, that is census data from England and Wales in 2001. And this data is available online at a range of different resolutions. In this case, our meta-community comprises 8,850 cast wards, and these are our sub-communities across 80 age groups. And we define similarity as a transformation of the distance between these age groups. In this case, we're looking at one of our beta diversity measures, representativeness, to find the 10 cast wards or sub-communities that have the least representative age distribution of England and Wales. And we find that these are predominantly university cities, such as, for example, Oxford University, Keele University, and the University of York, but also retirement villages, such as this Victorian seaside resort in Highcliffe, are very unrepresentative of this data set. We can conduct a similar analysis on a comparative data set where age groups are more broadly defined, and we find that we get similar results despite these differences in resolution. Diversity measures are commonly used to quantify biodiversity, and so for our second case study, we look at the Barro Colorado Island forest dynamics plot. And this is a well-known data set, a fully sampled 50-hectare site. We partition it into 20 by 20 meter quadrats and define similarity in the naive sense such that species are completely distinct from one another. And this gives us a better view of how our measures are operating. We select a number of our sub-community measures to pick out structure in different ways. And here we're looking at a measure of within sub-community diversity. This tells us that the effective number of species is varying considerably across the study site. No clear patterns are observed. Looking at a measure of between sub-community diversity, here is representativeness. And representativeness is low when a sub-community comprises a high abundance of species that are rare across the meta-community as a whole. And two regions are identified here as being particularly unrepresentative. That is, in the center, a swamp, and in the top left, two sub-communities of invasive species. Another measure of between subcommunity diversity is distinctiveness. We find that the swamp is actually moderately more distinct than the surrounding area, and this is because though it is unrepresentative, it has a low abundance of individuals. Whereas the invasive species in the top left are both unrepresentative and have a high abundance of individuals. We can also look at subcommunity gamma, which gives us the contribution towards meta-community diversity. And this is high when a subcommunity comprises species that have a low similarity to other species and are also rare across the meta-community. We can incorporate a measure of taxonomic similarity. And in this case, we are using parameters defined by Shimintani. And we find that the swamp is contributing highly to the taxonomic diversity of the meta-community, whereas the invasive species, having a much higher similarity, a taxonomic similarity, than the swamp, are no longer contributing. As well as looking at spatial diversity, we can look at temporal diversity by rearranging our meta-community such that our meta-community such that it comprises a subcommunity at multiple time points. And here we're using one of our beta diversity measures to look at the average distinctiveness of subcommunities through each point in time. We find that a high temporal beta is indicative of a high turnover in species composition over time. Looking at our spatial plots, comparing 1981 to 2010, we can see that the abundance of individuals increases in the swamp area, as does the representativeness of these subcommunities, agreeing with these observations. A third and final case study looks at antimicrobial resistance of Salmonella DT104 in a sympatric population of humans and livestock. So, in a previous study by Alison Mather and colleagues, they used Bayesian phylogenetic diffusion models to reconstruct the host population of branches in a phylogeny. And they found out that the majority of branch-to-branch transitions occurred between human and livestock branches, with a minimal amount of interhost transition. We took that phylogeny and applied our beta diversity measures, in this case the effective number of phylogenies. When the effective number of phylogenies is equal to 1, that means our human and our livestock phylogenies are well mixed. When it equals 2, that means our human and livestock phylogenies are distinct. And as Q increases, our estimate of the effective number of phylogenies is becoming increasingly conservative. There should be a grey bar there, but it's not. That indicates the null distribution, and it should show that at almost all scales, our phylogenies are not well mixed. And this depends on where we set the cutoff and how much evolutionary history we want to incorporate into our measure. We find that if we focus in on the epidemic, then the effective number of phylogenies increases, agreeing with the previous hypothesis that the animal and human epidemics are distinguishable. We can conduct a comparative analysis using genetic sequence data, and we get similar results. And again, this can be fine-tuned depending on how we define similarity. Alternatively, we can conduct a phenotypic analysis, in this case using data contributed again by Alison Mather. Our meta-community comprises 65 unique resistance profiles, where a resistance profile is a 13-digit binary string, each element of which denotes the presence or absence of resistance to a particular antimicrobial. And here we split our data set into 6-year chunks to allow a fair comparison through time. Each 6-year chunk is a meta-community partitioned into 3 sections, 1 emerging resistance, and 5 years prior of human resistance profiles, 5 years prior of livestock resistance profiles. And we want to ask the question, from which prerequisite population, from the animals or the livestock, is emerging resistance originating from? And so we use one of our beta diversity measures to calculate the redundancy and find that the human population has a much higher redundancy than the livestock population, telling us that the human population of resistance profiles is most similar to emerging resistance, and suggesting that it is unlikely that antimicrobial resistance originated from the animal population. So to conclude, we have applied our framework over a wide array of problems, over multiple fields of study. This framework is robust yet sensitive. We can alter our parameters to look at taxonomic diversity, phylogenetic diversity, and so on. We can alter our Q parameter that An Chao very nicely explained this morning to make our measure more conservative. Our measures are easy to use, often simpler than traditional approaches, and we also have a package available in R where questions and comments are appreciated. Thank you. No? Great.
You presented a variety of different approaches that can be used in. Is there any way that you, or anywhere in particular that you are hoping to use it in your group for a particular problem?
Particular problem, no. I guess we're just looking to explore it in different ways. Most of my work is kind of theoretical, computational simulations.
Okay, and so do you think that there's lots of different measures and tools for measuring
Yes.
diversity, and lots of different indexes and measures, and that came up in this morning's talk. So do you think the utility of this is because it's in one package that has many different levels of tools, or is there something new about this that's different to other tools that we've used in the past?
I think the generality is very important, and in this instance, we are able to partition diversity at the sub-community level as well as for a single population.
Okay, thank you very much. Thank you.
Cheers. No. Oh, okay.
So, our next speaker is Amanda Trash. She's going to be speaking to us about red-billed chaff population, in particular assessing the genetic threats to small populations.
Hi, so yeah, I'm going to talk about estimating effective population size in age-structured populations. Effective population size is important for understanding evolutionary processes in populations, and it's also important for assessing the viability of populations of conservation concern. So first of all, effective population size is the size of an idealized population that would experience the same rate of genetic drift as the focal population. And by an idealized population, I mean a right Fisher population, which is one of constant finite size, and where there's random mating between individuals and no migration or selection. So basically, the only force acting on this population would be genetic drift. So effective population size can be measured using both genetic or demographic approaches. The kind of genetic approaches which I'll focus on here are the single sample approaches. So I'm going to just do a quick comparison between those kinds of approaches. So the genetic approaches usually estimate inbreeding effective size, so they will estimate effective size for the parent generation. And this means they'll actually lag behind any changes in population size by at least one generation. So this could potentially be a problem if you're estimating effective size in populations that are changing in size. Demographic approaches, on the other hand, they'll estimate effective size, they'll estimate variance effective size, so they'll estimate effective size for the offspring generation. So this means that they can give you a more current estimate of effective size. The genetic approaches will estimate effective size from the resultant effects of effective size on genetic variation in that population. So this will mean that they usually don't inform on the underlying processes that gave rise to that effective size. However, because they estimate effective size from all those resultant effects, they will still capture the genetic effects of immigration into the population, which may otherwise be unobserved. The demographic approaches, on the other hand, they estimate effective size from the causal demographic parameters. So this means that they can inform on the underlying demographic and ecological processes that gave rise to that effective size. So that can be quite useful from a population management point of view. However, these demographic approaches rely on strong assumptions of discrete generations and stable age structure. So really, there's a need to incorporate, there's really a need for a demographic approach which can incorporate complex age structure into the effective population size estimate. So just such an approach has recently been developed by Engen et al., and this approach incorporates overlapping generations and complex age structure using Fisher's reproductive value. So Fisher's reproductive value can be defined as an individual of a given age's contribution to future population growth rate. And it's this distribution of individuals' reproductive values within and between years which can then be used to calculate total population demographic variance. And it's this total population demographic variance which can then be used to estimate effective size, along with estimates of the census population size and generation length. However, so far, this Engen approach has only been implemented as a methodological example and in a meta-population of house sparrows, considering only 2 age classes. So really, there's a need to implement this approach in a closed population where the effects of gene flow aren't going to obscure the effects of local demography, but also in a population with detailed enough demographic data so that we can actually calculate this total population demographic variance. Luckily, just such a population exists. So this is my study system, red-billed chough in Scotland. So red-billed chough are a species of European conservation concern. In particular, in Scotland, they're threatened. There's only 2 populations remaining now. And the population which I'm focusing on is the population on Isla, where there's about 46 breeding pairs. And because of this population's small size, and we know there's low genetic diversity there, so there's potential genetic concerns. So we need to quantify these genetic threats by estimating effective population size. So the red-billed chough on Isla have been the focus of a long-term study where the offspring are colour-ringed, and then that means we can follow the adults and work out breeding success and survival. However, red chough are a long-lived species, so there's lots of overlapping generations there, and there's also age-specific reproduction and survival. So really, we need this Engen approach to be able to calculate effective population size. I'm only going to briefly go through the methods here. I'm not going to talk that much about them. So if you want to know more about them, then please come and find me at the end or ask a question or whatever. But briefly, the demographic variance was estimated from samples of individuals between 2007 and 2014. And this approach has 2 levels of data to it. So first of all, the population level means. And here we used a 5-age class, 2-sex model. And we used 5 age classes to capture the age-specific differences in reproductive success and survival, but to also maintain sufficient sample sizes within those age classes. And we estimated probability of survival, probability of attempting to breed, and also breeding success of those individuals that attempted to breed. And this all went into a population projection matrix, which considers female and males' contributions to female and males of the next generation. And then the second layer of data that we needed was this individual level variation. So here we looked at realised survival of samples of individuals, again with this 5-age class 2-sex model, in their survival to the following year, and then also their breeding success. And this was needed to calculate mean sex-age year class demographic variances. So first looking at the population level data. So on the top you can see breeding success, and on the x-axis is the age classes, 1 to 5, with the 5-age class being 5 or older. And these in purple is the females and blue is males. And you can see breeding success increases from age 2 to 3, and then it's a bit lower for age 4, and then increases again. But this is breeding success of those individuals that actually attempted to breed. So on the bottom is probability of attempting to breed in the purple and blue again. And you can see this probability of attempting to breed increases with age until you get to age 4 or older, and then all individuals attempt to breed. And then in red is our probability of survival. And here we use recapture models to estimate this. And we found the best supported model was one where there was 3 age classes. Probability of survival starting off pretty low for age class 1, but then increasing to age 3, and then individuals get this adult survival probability. And here we, previous analysis found there's not really much difference in survival between the sexes. So we had, we put all the data into the model there. So we just had the probability of survival equal between the sexes. And this went into our population projection matrix and gave us the reproductive values and stable age distribution. So reproductive values, as expected, they increase with age, and the males have slightly higher reproductive value than the females. And then our stable age distribution is equal between the sexes because we have this equal survival probability between the sexes. And then looking at our individual level variation, and first looking at breeding success of the individuals. So here it's important to be looking at the variance within each of these age classes. So for example, you can see with these older age class males, where a lot of these individuals are actually having 0 breeding success, but then some of them are producing 4 offspring per breeding attempt. So there's a lot of variation there. And then also looking at the proportion of individuals that survive to the following year. So this is slightly different to what I just showed you on the previous graph, which was probability of survival, which was equal between the sexes. And here you can see there is some slight differences. But this is realised survival of a sample of individuals, so there's going to be some sampling variance. And these population level means and individual level variance then went into our estimate of total population demographic variance. And we find it's pretty high for the Isla-Chuff population. It's about 0.71. And when we look at the sex-specific contributions to this demographic variance, we find actually they're fairly similar between the sexes. But when we dig a bit deeper and look at sex-age-specific contributions to demographic variance, this is where it gets pretty interesting. So these younger age class individuals, the females seem to be contributing more to this demographic variance than the younger age class males, but then this pattern is reversed in the older age class individuals, where males seem to be contributing more to demographic variance than the females. So this suggests that there are key sex age classes which are contributing to this demographic variance and which are affecting the effective population size. And because there's this high demographic variance, then goes into our estimate of effective size, and we get really a very small effective size of only about 30. So already we were expecting a small effective size because there's only 46 breeding pairs, but this is really a critically small effective size because of this high demographic variance in the population. And then we wanted to compare and contrast our demographic approach to some genetic approaches. So we DNA sampled adults in the population between 2007 and 2014, and we had 2 different genetic approaches. So we used linkage disequilibrium, which measures association between alleles at different loci compared to expectations under random mating. And we also used approximate Bayesian computation, and this uses a variety of different population genetic summary statistics. And this is what we find. So first of all, in green is our demographic estimate of about 30, but then you can see from this linkage disequilibrium estimate, it's a bit higher, an estimate of about 50. And this is possibly because linkage to disequilibrium, this approach can be biased by processes in the previous generations. So it's possible that it's being upwardly biased by the previous larger historical size of the Isle of Chuff population. And then this approximate Bayesian computation approach, this gives us an estimate which is a bit more similar to our demographic estimate, so an NE of about 26. And this is possibly because this approach, because it uses a variety of different population genetic summary statistics, it might be being less biased by processes in the previous generations. However, using whatever approach, you still find this very small effective size, and certainly a lot smaller than the census size. And because of this small effective size, you can then estimate increase in inbreeding per generation in the future. And we get an estimate of about 2% increase in inbreeding per generation. So in conclusion, there's high total demographic variance in the Isle of Chuff population, and in particular there's key sex-age classes which seem to be contributing to this and acting to decrease effective size in this population. So this suggests that effective population size estimates should really aim to incorporate sex-age specific demography to provide unbiased estimates and also elucidate these key underlying processes that give rise to the effective size. The Isle of Chuff population has a critically small effective size of only about 30. And there's no evidence of unobserved immigration into the population because our genetic approaches also showed this small effective size. And we're expecting rates of loss of genetic diversity and increase in inbreeding per generation to be high for this population in the future in the continued absence of immigration. So just to conclude, now what we really need, our next challenge would be to incorporate these genetic threats alongside the ecological threats to the population and try to build a model for that so that we can come up with management plans in both the short term as well as in the long term for this population. So thanks.
Thank you very much. And any questions, please? I have a question for you, and people still thinking. So you said that you've been able to identify particular groups that are contributing to the demographic variance and the effective population size reduction more, and it was like older males are having a big contribution. So does that mean that, putting the idea that the population size is pretty small, but if the population size is the biggest threat or the inbreeding is the biggest threat, what can we do to these older males to stop them getting most of the reproduction population?
So probably, we think that this demographic variance might be linked to underlying variance on Isla, where like some territories are like really productive and other ones aren't. So it could be linked to that, because we already know, that in some areas on Isla, the pairs produce a lot of offspring, and also especially because Chuff, they're territorial and they'll stay on that nest site over over continual kind of years. So individuals that are doing well will keep doing well. So it could be linked to that. So actually, we still need to manage the habitat and make sure these poor quality habitats are improved. Yeah. So that doesn't seem to be the case because some of my previous work actually found that there's this emergence of a lethal allele in the population causing blindness. So we know definitely that there are still some lethal alleles in the population, that seems to be increasing with increasing inbreeding in the population. There's also seems to be some increase in, well, I don't know about increase, but there seems to be a fair amount of parasites in the population at the moment, and there isn't really any good data on that to see whether it is increasing or what's going on, but these are all kind of the sort of things that I usually people usually associate with inbreeding in the population. Yeah.
Thank you very much.
Okay.
So our next and final speaker this afternoon is Georgina Palmer, and she'll be speaking to us about biological responses?
Hello. So I'm a postdoc at the University of York, and a lot of my research is looking at describing species responses to climate change and understanding why there's so much variation among species in their responses. So, for example, a study that I was involved in last year looked at the northern range margin shifts of nearly 1,600 British species from 21 taxonomic groups across the UK. So on this plot, anything above 0 indicates that the species in those populations have shifted northwards across the time periods that we were looking at, so over the past few decades. And as you can see, on average, species have been shifting northwards, but there's a lot of variation both within taxonomic groups and across taxonomic groups. So my research is trying to understand why there's so much variation among species. And one of the hypotheses that I'm going to go into now is looking at the role of extreme climate events in driving variation among species. So commonly, the approaches to this sort of research identify an extreme climate event. So for example, in the UK, there was an extreme drought in 1975, and then look at species responses in that year or in following years and try and link the two in that way. However, I'm going to take a different approach. So I've got a long time series of data for many species in the UK, and I'm going to use those data to identify, first of all, which years are extreme for each species individually, and then try and link that back to the climate that's occurring in those years. So I wanted to answer 3 main questions. First of all, do responses to extreme climate events differ among species and among taxonomic groups? Secondly, are extreme population responses associated with unusual climate events? And finally, do extreme species responses drive the long-term population trends of species? So this work is focused on over 200 species of Lepidoptera, which are mainly macromoths, but there's some butterflies in there too, and bird species. And for all of these species, we've got data from either light traps or transects from these sites across the UK, from the 1960s to pretty much the present day. And we can use those data to generate national indices of abundance, an example of which is shown here. So in each year, we've got an index of abundance for each species, and we can use these data to calculate the long-term population trend. So this species has had a long-term population decline. And we can also use these data to identify extreme biological responses, which I'll go into next. I've also got a time series of climatic data. So I've derived 7 climate variables from the UK Met Office data, which we thought would affect the species that we were looking at. So these are measures of rainfall, drought, temperature range, growing degree days, coolness and hotness. And as I said before, I first of all identified extreme population changes and weather events, and then looked for links between the two. So first of all, to identify extreme population changes, I took those national indices of abundance, which I showed you on the previous slide, and then calculated the change in index for each species each year, between each year pair. So anything above this 0 line indicates population growth from year to year, and anything below this 0 line indicates population decline from year to year. And this is an example for the mottled grey moth. We identified extreme population changes for this species as these 2 years here, and these were years where the values of population change from year to year were larger than 2 times the median absolute deviation from the median. So this species had a large population crash in the late 1970s and in the late 2000s. So we did this for each species in turn. So for example, for the tree sparrow, there was 4 extreme population crashes in the 80s and 90s, and 1 extreme population growth here in the late 2000s. So as I said, we did this for each species, and then we summed the number of species experiencing extreme population changes in each year of the time series. So on the in each of the graphs on the next few pages, we've got Lepidoptera on the left-hand side, birds on the right-hand side. We decided to analyse those separately because there was a correlation between the proportion of species experiencing population changes for the butterflies and the moths, but not for the birds. So it indicates that there's either different drivers driving their population changes, or the same drivers but with different lagged effects. So on these plots, we've got the proportion of species experiencing extreme population changes on the y-axis and year across the bottom. And as you can see from the distribution of these bars, species generally didn't agree about which years that they thought were unusual. So across all species, about 90% of species experience at least one extreme change during the study period, but every year was unusual from the perspective of at least 3 species. And in each year, well, for 41 of the 44 years that we were looking at, populations of some species crashed while others exploded in the same year. However, I'd like to draw your attention to a few what we term consensus years. So these are the years marked with labelled with the year underneath. So these ones here and here. So we've got 5 of them for Lepidoptera and 1 of them for birds. And these are years where there's an unusually large number of species that are being affected in that year, either a population increase or extreme population decreases. So, for example, for the Lepidoptera, the largest consensus year was 1976 to 1977, and this was when about 30% of species experienced population crashes in that year. And similarly for the birds, 1981 to 1982 was the largest consensus year, when about 30% of the birds experienced extreme population crashes. But what I'd like to draw your attention to is, even in this largest consensus year for both birds and Lepidoptera, only about 30% of species experience extreme changes. So most species, even in those extreme years, didn't experience extreme population change. So in order to answer the second question, are species responses linked to extreme climates? We first of all identified which years were extreme in terms of the climate. So for example, we identified that 1975 to 76 there was an extreme drought. In 1981 to 1982 there was a cold winter. And then we wanted to look at whether extreme population changes for all species, so not just the consensus years in this analysis, were linked to drought, cold, or climatic unusualness. And we identified climatic unusualness using a PCA of the climate data. So years that are further away from this origin of this PCA indicate that the climate was more unusual in that year. So these plots show you the proportion of species experiencing an extreme on the y-axis, and either climatic unusualness, drought, or cold, with no lag or with 1 year's lag. And as you can see, it was quite a messy data set. We did find 2 significant correlations, so between drought and proportion of species experiencing extreme for the Lepidoptera. So in years that were more extreme drought, the populations tended to undergo extreme population crashes in the following year. And for the birds in this plot here, colder weather resulted in larger number of population crashes. But these both of these relationships were driven by the most extreme consensus year. So when we ran this analysis without that consensus year in, the relationships fell away. So we found that there was an overall lack of association between climate and population responses, looking at all extreme population responses. But then we wanted to look at whether the consensus years, whether they were linked to extreme climates instead. And we did find that there was a positive association between population consensus years and extreme climate conditions. So in years in those 6 years where there was the extreme population crashes or explosions, there tended to be extreme climate conditions in those years too. Finally, I was looking at whether extreme responses are linked to long-term population trends. And again, we've got the plot for the Lepidoptera on the left and birds on the right. We've got long-term population trend for each species on the y-axis, and then the maximum population extreme on the x-axis. So anything within these yellow boxes indicates the species for which the largest population extreme was a crash. Anything in the purple boxes indicates species for which the largest population change was an explosion. And again, it was quite messy. For Lepidoptera on the left here, we found no association between extreme population change and population trend. For the birds, we did find a significant relationship here, but there's a small number of data in that analysis. So birds that had a positive, the birds experiencing explosions tended to have a more positive long-term population trend. However, regardless of the direction and the magnitude of the extreme, some species showed extreme population increases and others showed long-term some species showed long-term increases and others showed long-term declines. So just to conclude, we found that responses to extreme events did differ among species and among taxonomic groups. So there's highly individualistic responses. So each year was associated with extreme responses of a few species, and some species showed extreme increases and some showed extreme decreases in the same year. We then found that consensus years, so years where a large number of species were either crashing or exploding, tended to be associated with also climatically extreme years, so particularly cold winters and hot, dry summers. And finally, we were looking at the link between extreme species responses and long-term population trends, and found that rare single extreme events have a limited impact on abundance trends, especially for Lepidoptera. So this could be other factors such as gradual climate changes and non-climatic events such as agricultural intensification impacting on the populations. So, that's it. Thank you. It was just the national scale. So we didn't have... Yeah. So on this study, we just looked at the national England trends, but it would be interesting to look at site level stuff.
Have you looked at a number of years before? So you're talking about the effect in a particular year, but the effect the weather had a few years ago on certain populations. So
yeah, so for the Lepidoptera, it seemed that drought in the previous year tended to have a knock-on impact in the following years. We did look up to 2 lagged effects 2 years previously, but it just seemed that the 1 year for the Lepidoptera, and it seemed that the effect on the birds was had no lagged effect. I'm sorry. I haven't yet, so this is just hot off the press at the moment. So next analysis could be looking at traits. Yeah.
So a variation of some of the questions asked is that you looked at it in any 1 year, and I grant you that you looked at, maybe it's some gradual process that isn't climate related. But what about slightly less extreme events but more cumulative? So the idea that an event could occur over 1 year and another year and another year again in a row might have a build-up of effect that's maybe more related to trends than the extreme in any one event.
Yeah, so we did look at, there were some years where there was an extreme drought, for example, extremely good year, what we would term a good year, and then the following year they crashed even though it was the same weather conditions. I think, and the flip side of that. So it's not something we looked at specifically for this, but